Good day, fellow humans. It is us, Victor and Irysad, and today we'll be presenting about an article regarding cloning, the science involved behind it, its possibilities, as well as its complications.
The article that we have analyzed is called Cloning Humans: Biological, Ethical, and Social Considerations. Published and approved on the 11th of March 2017, written by Francisco J. Alaya of the Department of Ecology and Evolutionary Biology and edited by John C. Advice, both from the University of California. The term of cloning, or the act of cloning, deters from the Greek word of klon, which means twig in our modern English. The act of clone was predated and by the process whereby a new plant is formed using the cuttings from another. This were to say that the act of cloning was the act of producing genetically identical individuals. Complete cloning of a human species has been more theoretical than practical, but a German scientist by the name of Hans Adolf Eduard Dress began studying and researching about reproduction in 1885, where in 1902 he was able to create a set of twin salamanders by dividing an embryo into two separate embryos.
It should be known that individual human cloning is already happening naturally through the birth of twins and in some rare cases triplets. This occurs when one single fertilized egg grows into two individuals, resulting in their genetic sequence to be identical, and hence their similar likeness. However, the cloning of singular genes or DNA segments, if needed to be done at a specific time, cannot happen naturally, and is already being carried out in many genetic laboratories worldwide, using a vast array of technologies that are already several decades old. The science contained within this article is about human cloning, which is often referred to as therapeutic cloning due to one of the processes that duplicates embryonic cells to allow for organ transplantation or repair injured cells, such as the nerve cell. Cloning generally occurs through SCNT, which is an acronym for somatic cell nuclear transfer. This is the process that is usually applied in cloning animals, following a few steps, which is as follows. Number 1. Remove or neutralize the genetic information of the egg within the female. Number 2. Somatic cells are extracted from the individual, while the cell nucleus is transferred into the host oocyte. The cell nucleus is where the genetic information is stored. And number 3. As a result, the egg becomes fertilized, causing it to be stimulated such that it can begin development. This also ties back to the unit about genetics, as the cloning can only be achieved through the retrieving of the somatic cell and cell nucleus, with its genetic information into a host oocyte, which is an immature egg cell, and development can begin. Genetics talk about DNA sequences and how traits are achieved, as well as how babies develop from a fertilized egg.
This discovery is considered significant as it could potentially lead to the successful cloning of an individual human body with complete organs and working functions. But as of right now, a human individual cannot be cloned. What is cloned are the genes of the individual, or more specifically, the genotype, not the phenotype. In fact, replicating or cloning the genotype is already a strenuous and challenging task to complete. So it is unimaginable how long it will take to be able to successfully clone a phenotype. There are many obstacles and technical difficulties that are considered immense, even when the cloning process for a genotype. So being able to clone an entire human individual is long and gruesome road to take, which would take several decades or even centuries to be able to perform such a process perfectly. All in all, many scientists believe that achieving a full human cloning is not impossible, but rather very unlikely, and it would take long periods of time to do so. This discovery is considered significant as it could potentially lead to the successful cloning of an individual human body with complete organs and working functions. But as of right now, a human individual cannot be cloned. What is cloned are the genes of the individual, or more specifically, the genotype, not the phenotype. In fact, replicating or cloning the genotype is already a strenuous and challenging task to complete. So it is unimaginable how long it will take to be able to successfully clone a phenotype. There are many obstacles and technical difficulties that are considered immense, even when the cloning process for a genotype. So being able to clone an entire human individual is long and gruesome road to take, which would take several decades or even centuries to be able to perform such a process perfectly. All in all, many scientists believe that achieving a full human cloning is not impossible, but rather very unlikely, and it would take long periods of time to do so. Be would be called as trying to personify oneself as a god, and many religions would call this sacrilegious to their faith and would never want this to succeed at all.
Another ethical reason for cloning to have complications are as if you were to propose the act of cloning someone who had passed away, would be immoral, and the clone would not be the same as the person who has passed away, since you can't clone memories or experiences from others. So, there would be no point in cloning someone who had passed away. It can be said that cloning a person who has passed away for the sake of their loved ones in order to help them cope with the pain of grief. But that is also an ethical problem, as this would ruin the reason to live and have experiences as a human being, because there would be no point in doing anything in your life if you are going to get cloned in the future. This would ruin us humans, as the whole reason for humans to live and make connections with others is to be remembered. But if you are going to be replaced in the end by a clone of yourself once you pass away. When homicide detectives check a crime scene, they would check for the DNA of the surroundings and see if the murderer's DNA was around. But if cloning were to occur, this would ruin the method altogether. Since there would be multiple people with the same DNA, no one can be sure if either the clone was the murderer or the original was the murderer. Along these lines, the threat of identity theft would increase as there would be many people with the same looks roaming the earth, and since they look identical because of their DNA, it would be easy for them to steal each other's identity and no one would know. The threat of online identity theft is already a big problem in itself. So how big of a problem would it be if real-life identity theft occurs? There are still a few questions that arise from this article, such as: Number 1. Will they be able to fit into society knowing the fact that they are made in a lab? Number 2. When the clone becomes sentient, who would be their parents/family? Would it if it had DNA from multiple people? And number 3. When the clone is formed, would the clone be sentient in an adult body or would they be born in an infant's body? And that is our short documentary about cloning, its benefits, complications, and possibilities in the distant future.